Hello, welcome to Learn It from a Layman. Carl Christensen. I'm back with Cameron and Matt and Johnny and no Tim. So we'll have to do without that this evening. Today we're going to be discussing the basics of DNA. So as you could guess, the expert, the layman expert, well, not even a layman, Johnny's going to be discussing DNA, and we're going to be handling some of the layman questions that I've come up with. So, Johnny, your background, once again, I think the listeners all know you're a doctor, but you have a degree in biology.
Yeah, a bachelor's degree in biology.
Nice. So, with that background, then what?
That seems like so long ago.
That's right. Well, medical school makes you age very quickly, from what I hear. Okay, so let's just get right to the DNA stands for what?
Yeah, just a scientific word for the structure of it: deoxyribonucleic acid.
Deoxyribonucleic acid, right?
Yeah, it basically is just talking about the structure of what is included within DNA. So it has a ribonucleic acid, but it's missing one oxygen. So then you have deoxyribonucleic acid. And yeah, like that. I don't know how interesting this will be to our listeners, but there's several things that compose the DNA. So it's the deoxyribose, which is a sugar, and then there's a phosphate group, and then there's a nitrogen-containing base, and the base is what pairs together to make that cool double helix structure. So the bases from one strand and the bases with the other strand will pair together and make that double helix structure.
Okay. So just what I've got out of this, it's not named after a guy named Helix.
Correct.
Okay.
And it's not after Felix the cat, and somebody just spelled it wrong either.
Okay. Good. I'm glad. It was discovered by Crick. Is that right?
Yeah, so there's two.
No. The lady actually found it before Watson and Crick.
That is true. There is some. So basically, there's a way to be able to view very small structures in like a bigger space. They use crystal chromatography, I believe, where they will crystallize a structure, and then based on the way the crystal forms of that structure, they can use fancy math and stuff. And stuff means I don't really understand how they do it, but they will be able to delineate what the structure is at very small levels, like the DNA level. So this lady, Cameron, do you remember her name? I don't remember off the top of my head.
No, I'm trying to think, but she, did the
Rosalind
crystal
Franklin.
Yeah, Rosalind.
That's it.
So she was basically on this in this field where she was using the crystallized structures to figure out very small structures, and she had taught Watson and Crick how to do it. And I don't remember the entire story of it, but yeah, it's a little bit shady who was first and who wasn't. But Watson and Crick are mainly named for it, but she was very influential in the structuring of figuring out the structure of DNA as well.
Okay, and this was in the 1950s, correct?
I have no idea.
Okay, well, based on my quick Google search, the layman's wrench, the go-to tool, is the 1950s. So this is all pretty recent, which probably will give me some of the answers to the questions I'm going to ask you here in the near future. But pre-DNA, did we just not have any idea how cells did what they did?
So there were, my biology history is not
that's all right.
strongest, but there were some ideas about that there is some type of substance that controls what is passed on from one one person to another, or one animal species to another, or one plant species to another. But they didn't really figure out that it was DNA until later on, from what I remember. Sorry, that might be
no, that's good. We don't need to spend a long time on history. That's good enough for me. Just wanted some basic background.
I said X-ray chromatography, but it was actually X-ray diffraction that they used to figure out the structure of the DNA.
Oh, okay. All right. So we've got these strands of DNA. Where does DNA live in a cell?
So it depends on the type of cell. So if you're talking about our cells, like animal cells, DNA lives inside the nucleus. So the nucleus is like holds all the blueprints of the cell, which are contained within the DNA. And it has instructions on, how to carry out different functions in a cell. And certain things can be turned on, certain things can be turned off, but it's all held within the nucleus. And you have to be able to send those blueprints out to the cell to be able to do certain functions and make certain proteins and different machinery of the cell. And so that will get sent out of the nucleus as a copy, but it will be RNA. So it will have that oxygen on there. It's no longer deoxyribonucleic acid. It's ribonucleic acid. That's sent out of the nucleus. But the DNA is always contained within the nucleus, except at a specific time when a cell is dividing. Otherwise, it's always in the nucleus.
Okay, and then some process in the cell makes this RNA, which is a copy of the information on the DNA, and that's what gets transported.
Exactly. So
okay.
that is called transcription. So they're taking, one piece of language, and it's very similar. Like RNA is very similar to DNA, like the structures, but it's only single-stranded, and it's missing it has that extra oxygen group, and it gets degraded pretty quickly. It's not quite as stable as DNA, and it's more easily used than DNA. So that's why that gets used. And then that RNA will be translated into a protein. So that RNA will code for different amino acids in a certain specific order that will get put all together and make, a protein. And that's basically what your whole body is. Basically, the whole reason your body functions, why your cells run, is by various proteins and their functions and their different affinities for different molecules and all that sort of stuff. So proteins run the world. They really do. And they're very diverse, a lot of different kinds. And we could probably spend another podcast just talking about proteins.
Yeah. Basics of protein sounds like a muscle man type episode.
People would go for that.
Okay, the real question though, so when you're bitten by a radioactive spider, how does the information get copied into your cells?
So it's kind of like the COVID vaccine, and I'm just it's very controversial. That's what I'm trying to say.
Okay, all right. I mean, I've seen the beginning of Spider-Man. I know that that somehow that this spider DNA just gets spliced in, that because you see the double helix. Anyway, all right, we can move on past that.
Carl,
But
I know what it is. It's DNA helicase.
Yes.
That's how it gets in there.
Yeah, it's got the helicase.
Yes, DNA helicase. Because it
okay,
unzips your genes. That's what it
that's
does.
right. Okay.
It's your genes. Oh my god.
Okay. Okay. This is good. Now the inside the nucleus.
Who knew DNA could be so racy?
Okay, we're now back inside the nucleus. And what?
It's where it's safe. It's safe
Exactly.
inside.
Yes, if we're back. We've gone home. The is DNA all in one long strand? What's so we got a double helix, but is it stored in one strand? Are there multiple strands? How is it organized?
Okay, that's a good question. So remember how I was saying that different types of cells contain different types of DNA? I've mainly talked about human cells and DNA,
Right.
but something that's interesting to think about is also bacteria and viruses. Bacteria do not have a nucleus. Their DNA just is inside their cytoplasm. So it's just the cytoplasm is the jelly inside the cell that's inside the membrane, and the DNA just floats freely in there. In bacteria, the DNA is a continuous loop. They also can take in little tiny pieces of DNA as well and store them, and they can copy them and pass them on to each other. And that's one of the ways you get like antibiotic resistance is one um, bacteria will have what's called a plasmid or a small section of DNA that allows it to make a protein so that it is resistant to a certain antibiotic. And then it will pass that plasmid around to other cells. And when it divides, it'll copy that plasmid and put it in its progeny or the next cells that it makes. And those cells will be resistant to that antibiotic as well. So anyway, that's kind of an interesting aside, but
Is that how you get viral mutations as well?
yeah, so viral mutations work a little bit differently. So bacteria and viruses tend to mutate faster because they don't have the same type of machinery that we have to repair mistakes in the DNA. In fact, viruses, they don't have any, and that's what allows them to make such big changes and become different variations of the same virus very quickly. I mean, it's not like, like every year we have a different strain of flu virus because of the different various mutations that have happened within the genes. You don't see every year different types of humans coming out. so, there's some. So we keep the integrity of our DNA very closely, because when things start to go wrong with that, it leads to, like cells dividing out of control and things like that, which can be, which can cause cancer. So our DNA is really important to keep in certain structures, and it's very important to repair it while it's being copied to make sure that it remains stable. Whereas in a bacteria, they might divide every 20 minutes or so, and they are allowed to make more mistakes because they're if one cell line doesn't make it, then the next one can. So there's just so many of them, and they grow so quickly and divide so quickly that they can handle not having like a perfect repair system. Although some bacteria do have a good repair system, not quite as good as eukaryotes. Eukaryotes are like animal cells, and plant cells also will repair their DNA. But viruses, yeah, they like thrive on not copying things correctly, and the more messed up it gets, it doesn't really matter. There's going to be, a million viral particles that get released from a certain cell, and if even half of those aren't virulent, it doesn't matter to that virus. It's still made a lot of virulent babies, I guess you could call them.
Sounds like middle
Viral
school.
baby.
Yeah, well, no, this sounds like middle school math.
Yeah, exactly. If you have a penny and you double it every 1 second, isn't that better than having $100 every minute? Anyway, something like that.
Yeah. Well, I was just thinking
So,
middle school students throw stuff against the wall and see what sticks.
oh yeah, yeah. That too. And then, so you were asking.
Original question: how the strands of DNA inside the nucleus?
Oh yes. Okay, so inside like an animal cell or a human cell, the nucleus stores all the DNA, and it's stored in there in kind of a haphazard, like mess of spaghetti. It's organized in a way, though. There are certain proteins called histones that will help organize the DNA. So if you think like your skin cell has the same DNA as your brain cell, but those cells are very different in what they do. So there's going to be certain sections of DNA that's turned off, and there's certain sections of DNA that are going to be turned on. So depending on what's on and what's off will determine the type of cell that is being, made or is functioning in the way that it is. So these histones will tend to wrap up areas that aren't functional, and then leave the other parts that are more functional to be copied more freely. So you're able to compact this very complicated strand of there's just strands of DNA, uh, and keep them organized within the nucleus. So humans have 23 pairs of DNA called chromosomes. So you have 23 chromosomes that came from dad. I should say most people.
Right.
So and these chromosomes, you got 23 from mom and 23 from dad, and they're very similar in 1 through 22. Number 23 is the sex chromosome. So boys, or people that are born male, have an X and a Y, and people that are born female have an X and an X, and that determines the gender as the 23rd chromosome. And when your cells replicate, this loose DNA that can be copied and used and things like that become rolled up really tight, and condensed into a chromosome that you can actually see under a microscope. You can see the different chromosomes under the microscope, and they'll all line up in the middle, all 46 of them. Yeah, and then during cell division, they get pulled apart. So half will go to one cell, half will go to the other after all that DNA has been duplicated. So it makes an identical copy of the previous cell. That's the way it's called mitosis. If you've heard of that, that's the way mitosis works. You make a replica of the cell that is identical, basically a clone.
Right. Okay. Chromosomes were one of my questions. You said most people have 23 because there are genetic issues where people do have an extra chromosome, right?
That is correct. So there's that. I think the most popular one that people will know of is Down syndrome. So that is called trisomy 21. So trisomy means that you have an extra chromosome. So you should have 2 chromosomes, but trisomy means you have 3. Somy means body, so you have 3 bodies of the 21st chromosome. So chromosome 21 has an extra copy, and that causes, what we see as Down syndrome, where people have the the down slanting eyes. They have like a single palmar crease. Their ears can be like a little bit lower set, and they usually have developmental delays and things like that.
Okay, so that, like you said, that's one that I think everyone's familiar with and kind of understood that had something to do with chromosomes if they didn't have the specificity that you just gave us. There are probably other ones as well that maybe we don't know as well, but I'm guessing that some chromosomal issues would just result in cell death and just death, right?
Yeah, exactly. So chromosome 20, so they're the way that they're named, the chromosomes are in order of size. So chromosome 1 is the biggest. Chromosome Y, which is 23, is the smallest. X is bigger, but they just took the 23rd and made it X and Y. So 1 through 22 are really just ordered by size. So 21 is a pretty small chromosome. And when you copy a small chromosome, you tend to be more functional than if you copy a bigger chromosome. So there's nobody that's alive that's a trisomy 1 or 2 or 3 or 4, like those don't survive. The trisomy 21 is survivable, which is why you see that one a lot more. But these are just mistakes that are made during the replication, and it causes, there's a lot of different things that can come from it. And, we could get into genetics at some other time.
Sure.
Like there's an entire specialty based on genetics, and different,
okay. Right.
different things within that can go wrong in cell division and what that can cause and things like that. But some of the more popular ones that you might have heard of is like Turner syndrome, which is where you only have one X chromosome, no Y, and Klinefelter syndrome. Some of these are a little more prevalent in the community, although like I think there's a lot of people that live with these and we don't really know. We like, without being a medical professional, you probably have no idea that this person might have some type of syndrome. You may think that they're a little bit different, but you wouldn't really be able to put your finger on it until you study some of the different trisomies and unisomies that exist.
Okay. Yeah, that's all very interesting, and like you said, it probably could be its own podcast. Going back, so we've discussed all the organization of the DNA, and you said different chromosomes are larger and smaller. Just to give us an idea, though, so these DNA strands, these chromosomes, are made up of these amino acids. How many? So these are all encoded, like in the DNA. It's got all these different amino acids, right? There's 4 different types of amino acid in DNA, right?
Yeah, that's right.
And how many amino acid double pairs are there in like the first chromosome?
So like chromosome
It's like millions
1,
or?
Yeah, millions.
Okay.
Let's see. So I'm reading this. So chromosome 1 is the largest, and it's 220 million base pairs. So if you were to stretch it out in a line, it would be 85 millimeters long. So that's
Crazy.
pretty impressive.
Yeah.
Like this is a a macromolecule, but very, very strong and small nonetheless, and pretty strong in the way it's built and able to withstand things for a long period of time. I mean, it's pretty impressive how they're using DNA for like to solve different crimes and things like that. Like the DNA actually can stay stick around for a very long period of time, years and years, without being corrupted, just because of the way it's all formed. It's very interesting.
That's awesome. Yeah, it's mind blowing how all of these things work together, and just the size and the scale and everything. But so with all these, I guess before we move on with discussing DNA, just the idea that this is encoded information. We've talked a lot about encoding in this podcast before. I mean, binary as far as computers go. So this is just a different way to encode information, is with these amino acids. That's essentially what's going on, right?
Yeah, exactly. It's just a very poor coded system. So
Right.
if you think about it, like, I don't know how much, you probably know more stuff like this, but how much of the stuff on your computer is like never ever used, that's coded in within the system? Do you know off the top of your head?
Not off the top of my head, no.
But I mean, if you're like running a system that only uses 50% of like what's coded in there, that's probably like, pretty low.
Right.
But
Right.
DNA is like really, really low. Only 2% of the DNA is actually used to code for proteins in your body. So that means 98% of it is just extra stuff. And some of it, there's some theories that it's important um, for structure and things like that, but a lot of it just seems redundant and things that aren't really needed. So it's kind of crazy that there's that much of
Wow.
the DNA that isn't ever used. It's just sits around in your cells and does nothing.
I didn't. If I had known that, I'd forgotten. So that is really interesting. That will take me, I guess, to my next question. So when we talk about um, us sharing some like 98% of our DNA with like a chimpanzee or something like that, is it this non-functional DNA? Is that even something that we know?
So that is, they usually quote that in functional and non-functional. So it's really a way to say we're more similar than what we might actually be. So basically there's 98% that's like not being used and 2% that is being used. So you say that you're, 99% similar in DNA to a chimpanzee or something like that, but really like 98% of that isn't being used anyway. So it's like 50%, you know what I mean, of functional
Right.
DNA. And I'm sure that there's people that have studied it to where they've done some of these things where they think about which parts of the functional DNA are similar and things like that. But it's pretty impressive how much DNA in living organisms is very similar. Like the same type of machinery is being used for different types of animals and even plants and things like that. Like there's certain things that are very important to life that exist in every single type of species.
Mm-hmm. Okay. So a sibling, shared DNA between siblings. So I've heard say that siblings are genetic copies of each other. I mean, they're not clones of each other, but I mean, is it just the active DNA that's different in siblings, or is it, do you know?
Yeah, so that's interesting. That's an interesting question. So this gets a little bit into statistics, which is a little bit interesting as well. So when your cells get ready to make gametes are your sex cells. So eggs for females, sperm for males. So if a sperm had 23 pairs of chromosomes and it got together with another um, egg that had 23 pairs of chromosomes, you'd have 2 times as much chromosomes as you needed, and that would be inviable. So you got to pare down the amount of DNA that is in the sex cells. So you do this through something called meiosis. Okay. Meiosis is different because you start with a cell that has 23 pairs of chromosomes, and at the end you have a cell that has 23 chromosomes, no pairs. So when that one gets together with the other, if that sperm cell gets together with the egg cell and it has 23 chromosomes, now that cell has 23 pairs of chromosomes. Okay. So the way that this happens is you get the chromosome 1, the copy that you get from mom, and chromosome 1, the copy that you get from dad, will line up right next to each other. Okay. And one of them will be pulled into one cell and the other one will be pulled into the other cell. Okay. Then chromosome 2 lines up the same way. And one of them will be pulled into one cell, one of them will be pulled into another cell. Now, there's 23 of these. Okay. So it's, you have a 50% chance of getting chromosome 1 from your mom, and a 50% chance of getting chromosome 2 from your mom, and a 50% chance of getting chromosome 3. I don't think I'm explaining this very well. Okay, so let me start over. So you have a 50% chance when the chromosomes align. So when a person is making their sex cells, their mother's chromosome 1 and their father's chromosome 1 will line up next to each other. And the direction that they go, left or right, into whichever cell is being made, is done by random. Okay. So you will have a mixture of parental DNA within your sex cells. So you might have chromosome 1 from mom, chromosome 2 from mom, chromosome 3 from dad, chromosome 4 from dad, chromosome 5 from mom, and it's completely random of how that goes about. Okay. So even with that alone, the amount of combinations that you could have with 23 pairs of chromosomes in two different cells, because remember, you're getting 23 from one sex cell and 23 from another, is,
okay.
I don't remember what it is, but it's very big. Like it's like the chances that you'll be an identical sibling for a mom that's pregnant by the same father is like impossible, basically. And that doesn't include something called crossing over, where when those chromosomes are lined up to each other, mom and dad chromosomes in the middle, they actually swap some DNA. So you may have a chromosome 1 that has most of mom's DNA, but it has a little bit of dad's DNA as well. So the amount of possibilities that you can get from meiosis is astronomical. Like, there's no way that you can have an identical sibling unless you're a twin, which is basically a clone.
Right. Okay.
A maternal twin.
Right. Okay, that's really interesting. Now, this is probably also, so I did the Ancestry DNA test a year or so ago. I don't remember when it was. Maybe it's 2 years ago. The Ancestry DNA, where you I don't remember what it is, saliva. I don't remember what they extracted from me, and then they test it, and then they figure out what percentage of the DNA you're getting comes from various parts of the world, and then they can map that in, or they map it obviously from your parents' DNA through to the different parts of the world that the DNA comes from, and so mine shows that I'm, got a certain amount of Scottish and a certain amount of Danish and all that kind of stuff, and it shows me which parent it comes from based on their DNA. So we have all of this that it's pretty mapped out these days. I guess my question here is, so we've understood now DNA for 70 years, but mapping all this stuff out is only been done pretty recently, right? Mapping the human chromosome.
Yeah. So copying, even when I was in undergrad, so even a decade ago, 2 decades ago, copying the entire genome, it was like Nobody's going to ever do that. There's no way. It's too slow. It's not going to be fast enough. It's going to take too long. We don't have the machines to do that. It's not interesting enough. And now we do it all the time because the machines are, so much faster, and we're able to do that. The way that they map out the DNA to different regions is usually done by the random mutations in non-coding regions of DNA that have specific like repeats or something like that. They can see where a mistake was made, and maybe everyone in Denmark has a mistake in the same spot. So they know that all those people came from
the Danish don't make mistakes, Johnny.
Yeah, they do. Have you ever heard their language? Sorry, all you Danes. But it does sound like you have a potato in your mouth.
Oh, my word. Okay, well, you just lost us. Every one of the Danes that have been listening.
Yeah, now the Swedes and the Norwegians are going to really jump on this, though.
Oh, man, it's okay. We got more Swedes and Norwegians that listen anyway. So.
Oh, that's okay then. Okay. So they can map it to different areas based on the random mutations that are passed down from, parents to children over time. These random mutations can be tracked back to different lines of DNA, which is very, very interesting. I'd actually like to do my own to see how much Neanderthal I have. I think it's probably a lot.
Johnny, you're a doctor. I don't know if there were Neanderthal doctors, so I think your prefrontal cortex is pretty well developed.
Hey, by the time somebody had to break the bone for somebody to be like, I think I can fix that, right?
Yeah, but do you remember here like 100 years ago when they were still bleeding people because they figured that would help?
Yeah.
We share a lot of DNA with those people. So.
Yeah, we do. Yeah, that's really it's kind of crazy when you think about it.
Yeah.
The amount of knowledge that we have is pretty incredible.
Yep. Okay. Let me take you back to something you mentioned earlier in the podcast, but I think is really interesting. So, DNA mutations and cause disease, and in a lot of cases, and something that a lot of people are familiar with is cancer, and that's an issue with DNA, right?
Yeah, most of the time it's an issue with DNA. So, DNA codes for the RNA, and then RNA makes the proteins. Okay, then proteins control the whole cell. So you have certain proteins that will tell cells to stop dividing. You'll have certain proteins that tell cells to divide. You'll have certain proteins that turn things off. You have certain proteins that turn things on. And if you have, the wrong blueprints to make those proteins, they're not going to function. And they're just going to be chewed up and found ineffective. So if you have a type of protein that tells cells to stop dividing that gets destroyed, then that cell's going to keep dividing with out of control. And as they divide, cancer is interesting because usually it starts out as maybe one thing goes wrong and it starts dividing out of control, but as it divides faster and faster, it gets less precise with all that editing that we were talking about and all that fixing the material and making sure that it stays correct. And then you start messing up other proteins that are important for the cell cycle and regulating the cell and making sure a cell dies when it's supposed to, and like all these different types of things, and it gets more and more and more out of control. There's also proteins that tell cells to stay within certain bounds. They're supposed to stay in a certain area. A muscle cell stays in the muscle, and a skin cell stays in the skin. And the melanocytes, they stay in the skin. And when the melanocytes get into, crazy duplication mode and stop, obeying their boundaries, and they can go to different places. They can divide and travel up into the brain or different organs, and we call that metastasis, where a cancer starts in one place and then this these cancer cells divide, they get less and less precise on what they're doing, and then they're able to invade tissues, which is, obviously worse than a cancer that is contained.
Right.
But yeah, it's very interesting. All has to do with DNA and different mutations. And the people that are in oncology nowadays that take care of cancer patients, there's a lot of genetic testing that they do to try to figure out exactly which chemotherapy regimen is going to work well for their specific cancer.
Right.
And I think, I think we have a lot more information now than we know what to do with. Like, we can replicate the DNA and we can see exactly which mutations they have. But I think a lot of times we still don't know exactly what to do with that information. Like, we have it, but we're still behind on, how do we use that to our best benefit?
Right. Yeah. And so I know that people are starting to try to figure out how they can, make edits to DNA, and so the CRISPR technology, right? That's an example of where they're trying to essentially live edit DNA inside cells, right?
Yeah, and there are successful DNA editing things. Let me see. There's a type of, I think it's called spinal muscular atrophy. I'll have to go back and check myself. If anyone's listening to this that is a geneticist, I'm really sorry. But they're missing some parts of their DNA, and they're able to get those into the cells that are affected, and then the people have like a normal life. So it's very effective for things like, this is in the nervous system, and your brain and spinal cord and nerves, you're not really replicating those cells. So you get the DNA in, and or like like I was talking about, those little segments of DNA that the bacteria pass around, you get a virus to deliver one of those segments of DNA to all those cells in the body that do that thing, and they're going to make that protein, and then they're going to have a functional cell at that point, because they were missing that protein before, or a functional copy of that protein. So it works well for cells that aren't dividing. But cells that are dividing, you put in that little plasmid and it gets lost in replication. So like a skin cell, for example, that's not, that it's going to divide 1 million times in the next month. I don't know exactly how many times, but a lot. It's not quite as effective. So they're still doing some things, but it does look like a very promising way of taking some of these very these diseases that have terrible prognoses and making them much better, and giving people more of a normal life, just based on the study of the DNA, the RNA, the protein structures, and what can we do to get those into the cell.
Yeah, and that's all just like forefront of medical research right now, right? That's a lot of what's going on in oncology and things like that as well.
Yeah, exactly. Like, there's you can basically make a cell line that will target certain types of cells, like an immune cell, and they can put that in the body, and if it's targeting your cancer cells, then it kills the cancer cells. Like, there's things like that are always in the forefront, and they're very interesting, so a lot of people read about them. It's not something that like everyone's doing, and it's not something I have ever like seen done. I've seen some of these things done, but like a lot of the stuff that's super interesting, like CRISPR-Cas9, and that's all still in research labs that they're doing a lot of that stuff. It hasn't made it quite to the public yet.
Right.
So no Spider-Man yet, but soon.
soon. Wolverine.
So when is the point to when I can like 3D print my own body part and fix myself?
I don't know. That sounds awesome, though. I know that
Or
they've been
just
working on 3D printing. Like a big thing that I've heard about is in so there's two things in endocrinology. Diabetes, especially type 1 diabetes, where your pancreas isn't making insulin anymore. Can we make like a structure that will hold beta cells that we can just put into the body, that we can 3D print? And then also people that have had heart attacks and part of their heart dies, can we get new heart muscle to grow on a 3D printed scaffolding, and then use that as like a patch that would actually be like functional heart, things like that. So these types of things are really cool ideas that just aren't being used right now, but something likely in the future that will be more commonplace.
Okay, I've got two
right.
more questions. Oh, sorry,
Wait.
Cameron, did you have?
I have one more. Why can't I just grow a clone of me and just keep it around for whatever
You
I
can't.
want?
Did you say you can, Johnny?
Yeah, go ahead, Cameron. We could always use a few more of you.
As true as that might be. What?
Yeah, so there are some problems with clones, and a lot of it has to do with the way the genes are. This gets pretty complicated, but just the way the genes interact based on whether they come from mom or dad is different in the body. So cloning gets rid of some of those safety guards, or I don't really know exactly what to call them, but things that are structurally and important for the cell to keep on living normally and to have an individual that's functioning normally. Like that hasn't been really figured out exactly how to do that. So anyway, your clone might end up being, like a photocopy of a photocopy. Not quite clear.
We all know what that looks like. Okay, stem cells. Stem cells are different in some way, but I'm unclear as to why they're different. And is their DNA somehow more enabled?
Yeah, that's a good question. So you have different types of cells in your body, and some of them they call it undifferentiated, which means that they can become several different types of cells. And you have some that are very well differentiated, that have reached the final evolution within their chain of different types of cells that can happen, and they're going to be that cell forever and ever. Like a neuron isn't going to revert back into and be like, I'm tired of being a neuron. I think I'll just try muscle today. That's a good thing.
That's what happened to Barry Bonds, though, I think.
yeah, it wasn't the steroids. Oh wait, he didn't steroid. Wait, I don't know. No. Was it him or Mark McGwire? Who knows? So if you have, everybody has cells in their body that are more undifferentiated, that are back further on this chain of changes that a cell can go through to become a cell that's more concrete, that's not going to change anymore. And those cells are called stem cells. So the undifferentiated cells can become several different types of cells. Those are called stem cells. And, really, like, your body started as one cell, right? Like a sperm and an egg got together, and at one point, you were one cell. And at that point, that cell had the potential to become every single type of cell in your body. And it did. So it made a copy of itself, and copies and copies and copies. And after a while, the groups of cells start to function in a certain way and will go off and differentiate into some type of material, or some type of tissue. And then a different cell will do something different until you have a functional body. And there's a lot of very complicated signaling and migration of the cells and all these different types of things in embryology that can go haywire, which is why, not every baby is born normal, but we love them anyway. And yeah, so some stem cells are very interesting and can do a lot of things. The fear, a lot of people are like, well, gosh, my X, Y, or Z isn't working right, so why can't you just put a stem cell in and it can become that thing? So sometimes when you put a stem cell in, you can't tell it what to become, and it will become something else, or it's lost some of that ability to regulate itself, and it can become cancerous. So it's something that's a little bit more serious than just, let's try a stem cell. Has to be very well thought out and very carefully done.
Okay. I always thought they were just like, little superhero cells that are out there, saving the day.
Sometimes.
Sometimes. Okay, I had one last question for you. Before that, I think I said earlier in the podcast, I think you said it right. I'm sure you did, but my lack of understanding, I think I said DNA is made up of amino acids. It's just not made up of amino acids. It's made up of nucleotides, and the code, the order, the way that the DNA is organized, then that is how it then builds amino acids for the protein.
Yeah, exactly. So the DNA is made up of nucleotides, and I may have misspoke as well because they sound very similar.
Right.
And then the nucleotides will code. They have like little keys on them that pair up with an amino acid, and they basically tell you what order of amino acids to put together to make specific types of proteins.
Okay, I think you probably said that, but I was unclear. I think at one point I distinctly said amino acids make up DNA. So we try to get things right on Learning from a Layman, even if I'm the one saying them incorrectly. So one last question, Johnny, at least from me. So I guess this is general, also just a PSA. So you'd imagine that DNA seems like it's outside of your control, and so things like cancer seem like they're outside of your control, and obviously to some degree they are. It's not, your DNA probably predisposes you for particular types of cancer, but there are lots of environmental things that go into what happens to your DNA as well. And I think an example a lot of people know of is radioactive, being around radioactive waste or something like that. That's going to compromise the DNA inside your cells potentially. But there's more than just that, right? The things that you eat, the way that you treat your body also does influence what's happening inside with your DNA. And so you can be hurting your DNA in a way that will then result in cancer. Am I getting that more or less right, Johnny?
Yeah. I mean, if you do have radioactive waste for a brain like Tim,
which you can say now because Tim's here. Before Tim got here, we can't slight Tim, but Tim has now joined us, so now fair game.
After all the Marvel movies I've seen, I am pleased to have radioactive waste for a brain.
That confirms it. Tim is a Tim. So yeah, there's things in the environment that play a role in how your DNA is expressed, how it's repaired, how it's used, and the better you treat your body, the better your DNA will be able to take care of you and that type of a thing. And yeah, there are environmental things that can cause cancers. There are things that can cause your DNA not to be repaired in the way that it's supposed to be and can mutate. I think one of the biggest ones you could talk about is smoking. Smoking's been linked to lots of different types of cancers. Now, there are people that are very susceptible to lung cancer because of their underlying genetic code. But if they smoke cigarettes, I think that will, like more times than not, that can tip them over the edge. So you just, and a lot of women will get breast cancer in their life. A lot of men will get prostate cancer. And there's certain things that you can do to try to, try to keep your body healthy. And, there's a lot of benefits of exercise and eating, as eating a good diet and things like this that are still being studied. And we don't really know how it works, but we do know that those people have decreased rates of cancer and, depression and, all sorts of things, just from trying to keep your body healthy.
Right. Yeah. So eat your fruits and vegetables, and you could actually be helping your DNA.
Five a day. You heard it from a pediatrician.
There you go. Awesome. Okay. Well, that brings me to the end of my questions. Matt hasn't said anything in this podcast. Tim just joined us. Cameron's asked a couple here and there. Any final questions for Dr. Johnny?
Sweet. I guess I made it really clear.
That's right.
Yeah, I think there's nothing further that we can learn about DNA right now.
You've covered it all.
How do I get my kids to eat their 5 a day?
You can't. That's decided by your DNA. So outside of your control.
Is our moderator happy that he is not genetically tied to eating his siblings?
Yes. Yes, there's lots of genetic material that as families you share, and sometimes you wish you didn't share. But hey, you know what? I had always been told growing up that male pattern baldness came from your mother's father. And so you just look at your maternal grandfather, and that's your hair future. And I'm grateful to say that I, for all of my other faults, still have a full head of hair. And my grandpa, my mom's dad, was bald by the time he was like 24. So take that, DNA.
Yeah, I think it's a little more complicated than that. But
anyway, thanks, Johnny. I think it was a good introduction to DNA, and we will ask everyone to let us know if you have any other additional questions, and we'll be back again next podcast. Thanks.